Hi, Mr. Kedsting here. I'm going to model DNA replication here with this paper chromosome. Paper chromosome that I have here is a double-stranded molecule. Here's one string of nucleotides. The nucleotides are A, G, T, T, C, T, A, G, 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 T, A. And the other side is another string of nucleotides that are complementary to it. The A matches the T, so they're complementary. The G and the C are complementary. The T and the A are complementary, and so on, because their shapes match. Now, there's direction to the molecules here. Each nucleotide has a 3' and a 5' side. So this is the 3' side of this T nucleotide. This is the 5' side of the T nucleotide. Of the strand, this is the 3' end. This is the 5' end where you see the phosphate sticking out. On this strand down here, this is the 3' side. This is the 5' end where you see this phosphate sticking out. What I'm going to do is I'm going to demonstrate how this paper chromosome would replicate. So I'm going to declare that my origin is over here. Okay. So what's going to happen here is helicase, which is an enzyme that breaks the hydrogen bonds between the complementary nucleotides AT and GC. Helicase is going to zip down the line here. My finger is going to serve as a helicase, and it unzips the molecule, opens it up. Okay. Now at this point, I got them separated just far enough I can make everything work. Now, it's going to turn out that this is going to be the leading strand, and this is going to be the lagging strand. And the reason is the leading strand is the side or the strand of the DNA where 3' is at the origin start. The lagging side will be where 5' is at the origin start. Okay? So enzymes called DNA polymerase will actually bond to these nucleotides, and they will put new nucleotides in position. So the DNA polymerase, which is going to be my hand, comes in and it sees a T here, so it puts an A on it. It sees an A there, so it puts a T on it. It sees a G there, so it puts a C on it, and so on. I'll go one more, and it sees a C there, and it'll put a G on it. Meanwhile, on the other lagging side, the enzyme cannot do that. Instead, it has to work backwards. So in that case, what it has to do is, as helicase unzips the molecule, it tends to bond somewhere in, say at nucleotide number 4, it'll bond, but the enzymes always slide in the 5' direction. And this is the 5' direction. So once it's put a C with this G, it'll put a C with that G, it'll put an A with that T, and then it'll put a T with this A. Okay? Now, meanwhile, I'm going to go ahead and throw a few more nucleotides on the leading side. The DNA polymerase is on the leading side. Just keep moving down the line. A G goes with the C, an A goes with the T, a T goes with the a C goes with the G. Meanwhile, on the lagging side, things work in a similar pattern as they did before. The enzyme will actually come in several nucleotides, and it will actually put, say, a G onto this C. And then it'll put an A onto that T. Remember, it's working in the 5' direction. It'll put a T onto this A, and it'll put a C onto that G. Meanwhile, on the leading side, the T will go with the A, the T will go with the A, the G will go with the C, and the A will go with the T. The top side is now complete. The bottom side yet is not. At the end here, a T will be bonded to that A, a C with the G, an A with the T, and an A with the T. Now, before we're done here, there's a special enzyme that needs to bond together these two nucleotides here because they're disjointed. This little string of 4 was put on, and then this little string of 4 was put on, and these two can't bond together without the action of ligase. Ligase bonds this pair of nucleotides together, and then it comes down here and it bonds nucleotides down here together. Wherever, let's see, 1, 2, 3, 4, it'll bond these together. 1, 2, 3, 4, it'll bond those together right there. And that's it. Ligase is not needed on the leading side at all. It's only needed on the lagging side to bond together the disjointed little sections of replicated DNA that bump into each other. That's it. What we have here is a set of duplicate paper chromosomes that were made as a result of DNA replication.